Good afternoon. It's a pleasure to welcome all of you here in Missouri Auditorium and those who are watching on the web for the Wednesday afternoon lecture. And today we have a special treat of a presenter, Dr. Elaine Fuchs, who has done really remarkable work in understanding developmental biology, and particularly as it relates to the epidermis. Dr. Fuchs has a distinguished academic career. She got her undergraduate degree from the University of Illinois, her PhD in biochemistry from Princeton. After a postdoctoral fellowship at MIT with Howard Green, she went on to the University of Chicago, where over the next 22 years, she did remarkable work in genetics and cell biology involving the skin. In fact, many of the real breakthroughs in understanding genetic diseases of the skin were a direct result of her work. And she trained a number of other distinguished investigators, one of whom, Julie Segre, is sitting next to her this morning, and others of whom some of us have had the pleasure of working with along the way. So she's also been a wonderful mentor. In 1993, she moved to the Rockefeller University, where she now is, as the Rebecca C. Lansfield Professor in the Laboratory of Mammalian Cell Biology and Development, and she's an investigator in the Howard Hughes Medical Institute. She's an elected member of the National Academy of Sciences, also of the Institute of Medicine, received a number of really important prizes: the Dixon Prize in Medicine, the L'Oreal UNESCO Award for Women in Science, the Pisano Award, the Albany Prize in Medicine, and the National Medal of Science. Her work is really focused on understanding molecular mechanisms in the skin, and most recently, particularly in the area of stem cell biology, involving the epidermis and its appendages. You may have seen a really interesting paper she published back in September from her group on the use of RNAi to understand additional important issues that relate to skin biology and oncology. And she has already told me that she's going to present some data here today that has never previously been presented in a scientific forum. So get ready. I'm sure it's going to be an interesting ride. Please then join me in welcoming this afternoon's lecturer, Dr. Elaine Fuchs.
So, can everybody hear me? Okay, well, first of all, let me say that it's a real pleasure to be here and participate in this series. And what Francis didn't say is that actually he inherited my very first graduate student that I ever had in the laboratory when I was first starting. And my first graduate student did his postdoctoral studies in Francis's lab and has gone on for many years to be a professor at Duke University. So we do have this thread that goes way, way back. And I thought what I would do today is to present to you some of the work that my laboratory has been doing recently, but at the same time, I wanted to give this some perspective. And so my laboratory uses skin as a model system for studying stem cells in looking at questions of silence, action, cancer, and we do so for some very simple reasons, because this is by far and away the most accessible, abundant, and synchronous source that we have of stem cells in our body. All of the tissues that you see here, the epidermis in blue, the hair follicle, and the sweat gland are all derived from a common embryonic progenitor, and these cells express keratins 5 and 14, which are the first keratins that my laboratory ever cloned and characterized. In fact, I would say the first ones that I ever cloned and characterized many years back. Another interesting aspect about the skin is that in the adult epithelia, during homeostasis and wound repair, and certainly we subject our skin to more wounds than probably any other tissue of our body, that many of these tissues, the epidermis, rejuvenates every 4 weeks' time. You've got a brand new epidermis on your body surface. Your hair follicles, of course, if you're lucky, throughout your lifetime will not only undergo periods of rest, but also periods of growth. And the sweat gland actually has much less activity during normal homeostasis and wounding. But there's an enormous reservoir, in fact, reservoirs of stem cells that must accommodate this tremendous amount of homeostasis and wound repair. And then, of course, when we're talking about cancers, and particularly squamous cell carcinomas, we're not only talking about the most prevalent, among the most prevalent cancers worldwide, but also among the most life-threatening. And so for all of these reasons, skin is really the opportune organ system to be able to study many of the aspects that we're interested in generally in stem cell biology. And so if we go to the very beginnings of stem cell biology, actually it's not all that far along. Back in 1877, Ernst Haeckel coined the term stem cell, and at that time really referred to these as the fertilized egg, of course, that gives rise to all cells of the body, and that was the definition of a stem cell. This word was popularized by E.B. Wilson in the late 1800s, who, in his book, The Cell in Development and Inheritance, basically described the stem cell from which arise the germ cells. And so, again, these early definitions were developmental definitions for stem cells. People really didn't think about stem cells as being residents to tissues until the work of Alexander Maximov, who was a Russian cytologist. He fled during the Russian Revolution, came to the University of Chicago, and in the course of his cytology studies proposed that hematopoietic lineages come from a common progenitor, a stem cell. But it wasn't until now, another 60 years later, that there became experimental evidence for the existence of these tissue-specific stem cells when the work of Till and McCullough in studying bone marrow transplantations at the serial limit dilution level described individual cells which were capable of reconstituting the entire hematopoietic system of an irradiated mouse. And so this concept then really led open the field of stem cell biology as we know it today, particularly for adult stem cells. And it was another 15 years later that the very first stem cells were cultured and passaged under conditions where they did not lose their stemness. And this really came from the pioneering work of Howard Green, who developed this methodology, making the breakthrough that stem cells often rely upon their neighbors in order to be able to provide the necessary factors that are important in controlling their proliferation and their stemness. And so Howard Green made the use of a fibroblast feeder layer that then supported the growth and propagation of human epidermal stem cells. When my laboratory first started out, one of my graduate students began to work with three-dimensional cultures. This was back in the early 1980s, where we could pretty much from scratch recapitulate cultured epidermal cells under conditions where they could stratify and differentiate. So these systems worked very well. Howard Green went on to develop this methodology for one of the first stem cell therapies. We often think of stem cell therapies as something of the future, but here is now a 30-year success of one of these examples. And this was using cultured stem cells, where he simply took a section of the good skin of a burn victim, basically made sheets and sheets of cultured epidermal cells, and then engrafted them onto the patient. These patients' epidermis, in fact, give us a 30-year standing of saying what happened with all that propagation going on in culture. The skin of these patients never showed signs of cancers or perturbations in the skin epidermis, and that tells us that stemness can be maintained and propagated in culture under conditions where the cells do not acquire serious deleterious mutations that would compromise skin behavior. We do know, however, that the skin of these patients does not sweat, nor does it make hair. And so that told us early on that there were other sources of stem cells that exist in skin. And one of the things that my laboratory has done is to systematically work our way to identify the resident stem cells of sebaceous glands, more recently sweat glands, also hair follicles. So the foundation of cultured epidermal cells also served as the foundation for the culturing of human embryonic stem cells and mouse embryonic stem cells. It was 5 years later that Gail Martin and her co-workers adapted what Howard Green had developed, the use of a fibroblast feeder layer, to embryonic stem cells, which are also epithelial-like in nature. And this then, of course, opened the door for mouse genetics, for human embryonic stem cells, and now for iPS cells. So a variation on the theme came 10 years after Howard Green's discovery from another postdoctoral fellow of Howard Green's laboratory, who made the realization that you could take corneal epithelial cells if there was a patient that had only one blind eye from, for instance, an industrial burn accident. You could take stem cells from the good eye, culture those, and passage them. This was the blind eye before treatment, and this after treatment. And after 10 years, 100 patients who were treated in this way basically had their vision restored as a consequence of this stem cell therapy. So again, another success rate in the field of stem cells for regenerative medicine. So if we look at the biology of stem cells, what we've learned is that not only do stem cells rely upon heterologous neighbors, but in fact stem cells of most adult tissues reside in some kind of a niche. That's true for the hair follicle, the intestine, and the hematopoietic system. And another thing that we've learned about stem cells is that they typically exist in two distinct states: a hyperproliferative state, or a primed or an activated state, as well as a quiescent stem cell state. And depending upon the particular tissue and the needs of the particular tissue, we find different degrees of quiescence and different degrees of primed or activated states. And so another thing that is important in considering stem cell biology is that in going from one state to another, from a quiescent state to an activated state, there has to be a change in the microenvironment. And so if stem cells are in niches, there have to be a change in the signals that come from those niches. That change in niche signals can come from many different inputs. That leads to an activated stem cell, which then typically gives rise to a short-lived progenitor or a transit amplifying cell that then goes on to differentiate to make the bulk of the tissue. So most of the tissue that you see is basically not coming from expansion of the stem cells, but rather expansion of their short-lived progenitors. So for me, studying this problem really made sense to study it in the hair follicle, because not only is the hair beautiful, but it really is the ideal for studying what how stem cells are turned on and what turns them off again. Because the hair undergoes these cyclical bouts of activity, and this is in normal homeostasis without any wounding, which might interfere with our interpretation. So in the normal state, the stem cells that I was talking about now are the red ones here. They're in a quiescent state known as the bulge, and that's in a resting state abutted up next to a mesenchymal component, the dermal papillae cells. After what can be even months sitting in a quiescent state, when the activating cues now of the niche are overriding the inhibitory cues, now stem cells from the very base undergo proliferation. They generate a transit amplifying population, now shown in green here, which, as the cells are growing downward and forming the new hair follicle, the dermal papillae, the mesenchymal stimulus, is being pushed further and further away from the niche. The niche returns to quiescence, and this is where the bulk of the activity for tissue development, in this case hair development, takes place. We don't yet know, but we've been studying lately, the destructive phase. All of a sudden, there's apoptosis that happens in the proliferative progenitors, and the entire system retracts until we get back to this stage again. And as I said, the resting stage then can last for weeks upon months. Unfortunately, our resting stage gets longer as we get older. And just recently, Bryce Keys in my laboratory has published a study on why that is. And again, it deals with this issue of balancing positive and inhibitory cues. So to be able to mark these stem cells and really study them in detail, we developed some years ago a pulse-chase animal that we could turn on a fluorescent histone and turn it off again. If we turn it on in embryogenesis and turn it off postnatally, what we find is that initially, before turning it off, all the skin epithelial cells have their nuclei with this histone fluorescence, and then if we turn it off by simply adding tetracycline to the animal's diet and wait 4 weeks, that's where we see this fluorescent population of cells. So they're relatively infrequently dividing next to their proliferative or differentiating neighbors. And so this allowed us a segue into monitoring the stem cells in their niche. As I told you, when stem cells get activated, they do so at their base. These are the label-retaining cells, now in green after that histone pulse chase. And as we start to look at the new emerging hair follicle in the next hair cycle, what we can do is to monitor the divisions by a factor of 2 dilution in histone H2B GFP. We can see that all of the cells in the emerging hair follicle are fluorescent. They just have less fluorescence due to the fact that they're dividing now, but they have more fluorescence than anywhere else except for the bulge region, which is the residence of these stem cells. So these data tell us that these quiescent cells, in fact, the cells at the base of the quiescent niche, the quiescent cells there, are basically the ones that are giving rise to this early new hair follicle. We now know that both these populations are contributing to the new hair growth. They are both in a quiescent state for weeks or months before getting activated. So now we can deal with the transition between stem cell quiescence to activation, because in the mouse, the initial hair cycles are synchronized. This allows us to look now with short pulses of nucleotide, in this case in green again, at what happens when now the new hair follicle is beginning to cycle. We see proliferation at the base, close to the mesenchymal stimulus, and those are the ones now that are proliferating and beginning to give rise to the new hair follicle. We also know that during this period of quiescence, where there's no proliferation whatsoever, what we see is that if we were to purify the bulge, the hair germ, and the dermal papillae, what we'd find is that the hair germ accumulates increasing levels of Wnt signals. The dermal papillae creates increasing levels of BMP inhibitors, and that culminates in nuclear beta-catenin as the fate to making a new hair follicle is made, and as these cells become stimulated at the base. So what this tells us is that these two signals, positive Wnt's and inhibition of BMP signals, are critical in driving the new hair cycle. It also tells us that even though there is quiescence, that there's a considerable crosstalk going on, changing the inputs that basically the stem cells are responding to, until finally the activating cues overcome the inhibitory BMP signals that are in the niche. We also know that if we get too much of a good thing, overactivation of Wnt's leads to cancer, underactivation of BMP signaling leads to cancers, as we discovered in creating various different of our knockout mice. So when we have these cells now fluorescently labeled, we can purify them and profile them, and we've studied quite a bit the changes that occur as stem cells go from a quiescent state to an activated state, to a state where they're fully committed to making transit amplifying progeny. And we know that relative to their progeny, that there are high levels of a variety of different transcription factors. Integrins are high. LGR5, a G-coupled protein receptor that is also implicated in intestinal stem cells, is 60-fold higher in these quiescent stem cells relative to their activated transit amplifying progeny. If we look at the niche, in the quiescent state, sonic hedgehog signaling is down, Wnt signals are down, and BMP signals and FGF18 signals are high. So there's these overriding inhibitory cues counteracting the activating ones that have to build up to tip the balance. What we also know is that if we were to knock out some of these various different transcription factors, as my laboratory has done, it turns out not to be quite so simple as what it seems in the embryonic stem cells, where all of the transcription factors are acting on pluripotency genes. In this case, if we knock out LHX2, what happens is that the entire stem cell niche basically turns out to become a sebaceous gland instead of a stem cell niche for the hair follicle. And what we know is that if we repress TCF3 or 4 signaling, or if that they basically are repressing Wnt signals, there's very high levels of these transcription factors expressed in the quiescent stem cells. There's very high levels of nuclear NFAT-C1, which is a downstream target of both BMP signaling and calcium. We know that they govern quiescence. We know that EZH1 and EZH2, the polycomb protein methylases, govern the switch between stem cells and transit amplifying cells. And we know that without SOX9, stem cells never form, and basically we don't get hair, and we don't get good epidermal repair, and we don't get sebaceous glands forming. So these seemingly very disparate functions for not only the various different transcription factors, but also for the various different epigenetic marks, is now leading us to wonder exactly how these regulatory circuits are controlling various different sets of genes. And so we're digging ever deeper into this problem. We have a real advantage here because we can isolate sufficient quantities of stem cells in quiescent state, activated state, and transit amplifying state to be able to do such detailed technologies as ChIP-seq analysis. And we're doing this in vivo in stem cells in their native niche as they're undergoing these transitional states. So another thing that I think is interesting that we often don't consider is that while Wnt signals play very important roles in a variety of different stem cells, that they don't simply act in the nucleus to change transcription. In fact, we've known for many years that beta-catenin is sitting out there at cell junctions, and we've only recently learned that actually when you lose beta-catenin at those cell junctions, there's not redundancy from other proteins such as plakoglobin out there, but actually there are real defects that arise in intercellular junctions. We also know that overactivation of beta-catenin can also influence cell-cell junctions. But now it goes even further. We know that there are also cytoplasmic effects, and now going to the other flip side of Wnt signaling, which is basically to block GSK3 beta kinase, there are many GSK3 beta kinase targets beyond that of simply beta-catenin. And we know that some of those can really have an impact on stem cell biology, as Xiaoyang Wu, who was previously in my laboratory and now at University of Chicago, has demonstrated through showing that there's one cytoskeletal regulator, ACF7, that is regulated by GSK3 kinase phosphorylation, and in the presence of Wnt signals, where the kinase is inhibited, now microtubules can be stabilized in a directional fashion, and that allows the stem cells to migrate properly into a wound site. So again, I think one of the reasons why we're finding so many different outcomes of Wnt signaling, depending upon whether we look, say, in the intestine, or whether we look in the hair follicle, or whether we look in a variety of different tissues, is that these factors and signaling not only are regulated by whether there's a positive or negative transactivator interactor for beta-catenin, but also by the variety of different GSK3 targets that are affected or impacted as a consequence. So another area that I think is interesting, and again, I'm trying to give you vignettes of what has emerged in the field over the last 10 years and how does our work fit into this, is that one thing that we realized very early on as we were purifying these stem cells is that if we look in their normal native state in homeostasis, these hair follicle stem cells will only regrow the hair follicle. That's what they do, and that's what they do in homeostasis. There are other resident stem cells that make these other tissues. But what we find is that if we purify these stem cells, taking them out of context, and we engraft them into a hairless mouse, we not only get these nice tufts of hair, but we also get epidermis and sebaceous glands, something that these stem cells do not do in normal homeostasis. So what is responsible for the difference? Well, based on what I've told you so far, I think you'll not be surprised to realize and reflect that there are two major differences that distinguish de novo morphogenesis from merely refreshing an existing tissue. One is that the stem cell niche is missing. We're putting this into a foreign environment, and if there are the other proper heterologous cells, those cells are capable of interacting with the stem cells and eventually reforming the niche. But it takes a while for that to happen, and a lot of things that can happen in the meantime. Another interesting consideration is the tissue progeny themselves are missing. And in fact, one of the things that Yache Su in my laboratory has learned is that in fact, tissue progeny can impact dramatically on the behavior of the stem cells themselves. So these are basically downstream signals that are emerging from the stem cell progeny. And that stem cells are regulated both by heterologous and progeny niche cells. And when you consider the whole aspect, for instance, of wound repair, where we get a wound, and of course we know that the wound can be repaired. Our stem cells get activated, they repair the wound. But you don't expect to see a tumor growing at the wound site. So there's something that's telling the stem cells to stop making tissues. Stop, you've repaired the tissue. And what better way to do that than to have tissue progeny downstream feeding back to the stem cells to say, okay, stop, I don't need any more of your activated cells. And in fact, we've discovered that differentiated cells can home back to that niche and provide BMP signals and FGF18 signals that further raise the threshold for a new round of stem cell activation. So this, I think, gives us some insights, and we can think about whether this applies to many different systems. I think that if you look at the literature over the last couple of years, there are now a number of emerging cases where it looks like this type of feedback mechanism is coming into play, both from the heterologous signals, but also from the autonomous signals. And just to give you one example, Iqbal Banerjee, who studies fly hematopoiesis, discovered that there's a downstream differentiation signal that feeds back to the hematopoietic stem cell in the fly that tells the stem cell to stop its activation. And so that's very similar to what I've just described to you here for the hair follicle. Another interesting aspect is that this system is perturbed in cancer. And in fact, you can think about cancerous tissue as now this system doesn't work properly. And when this system doesn't work properly, these signals aren't provided back. And in fact, we realized that several years ago. In fact, for every time we started to look at what turns stem cells on and what turns them off again, what we began to realize is that when we overactivated genetically that system, we ended up getting cancer. And when we underactivated the inhibitory cues, that gave us cancer. We began to realize that, in fact, tumor-initiating cells have pretty much hijacked the natural mechanisms that stem cells use to control their behavior to be able to control tumorigenesis. And so that led us to the question of where are the cancer stem cells and what do they look like? And several years ago, we tackled this problem by simply starting to systematically dissect out, FACS purify all the various different populations of the heterogeneous squamous cell carcinomas until we got down to, on some genetic backgrounds, at the single cell level in serial transplant assays, a single cell was sufficient to be able to introduce it into a host recipient mouse and get a squamous cell carcinoma that resembled that of the parent. And this could be done over and over again. That tells you that you've got a cancer stem cell, and then we wondered, what do these cancer stem cells look like? Well, like normal stem cells, they reside at the tumor stroma interface. So they too have their own niche. They express high levels of integrins, like normal stem cells, and they express high levels of self-renewing genes, like normal stem cells. And like normal stem cells, they exist in two proliferative states. There are some that are quiescent-like, or not as active as the other population. And in fact, we've even learned the difference between what controls the balance between those two quiescent states. In a squamous cell carcinoma, there's very high levels of TGF-beta that surround the squamous cell carcinoma, and as long as the squamous cell carcinoma cancer stem cells can respond to TGF-beta, that makes them less proliferative. If the cancer stem cells lose their ability to respond to TGF-beta signaling, now the number of cancer stem cells goes way up, goes up by a factor of about 10-fold, and we lose those differences between proliferative states of the two cancer stem cell types. But if we look at the similarities, that's about where the similarities end. In fact, if you profile the cancer stem cells, we find that there are more than 700 differences that exist between the cancer stem cell and either the hair follicle stem cell or the epidermal stem cell. So they really don't look anything like normal stem cells. Should we be surprised? Probably not, because now they've got their new niche. They've got a different stroma. They've got immune inflammatory cells. They've got different types of endothelial blood vessels coming around the tumor. So they have a different niche. They also have different progeny. They're going to have different signals, and they're going to have different properties. In fact, when we look at this, there are many different cancer genes. TGF-alpha is high. VEGF to recruit angiogenesis is high. What we find are the twist genes. Epithelial mesenchymal transitions is high. Kirsten RAS is upregulated. Cadherins, catenins are downregulated. This is a cancer signature list, but it's not a characteristic of normal stem cells. And so as we began to look at all of these different aspects, we began to realize that we can't systematically simply take a look at which of these 750 or some odd genes are really relevant. Can we take our conventional genetics approach? I don't think so. Certainly not to be able to go through the functional significance of all of these different changes. So this really became a challenge for us as we started to move to the human genome sequencing era and began to deal or try to grapple with all these hundreds of changes. If you look at the cancer sequencing genome projects that have been headed here for particularly head and neck cancers, the most life-threatening of all of the squamous cell carcinomas, and naturally there's been a lot of sequencing going on given the mortality rate, but there are hundreds of coding mutations per tumor occurring in more than 2 patients or more than 1 patient in these head and neck cancers. So they have the same kind of problem of how do you sift through the hundreds of mutations to find out which of these are actually drivers for the cancer and which ones are mere bystanders. Well, we can reference the 2 data sets. We can reference the human sequencing efforts, and we can reference our epigenetic changes, but we still get hundreds of changes. So let's compare the methods. Well, the exome sequencing doesn't even begin to identify epigenetic changes, but it does focus on human squamous cell carcinomas, and that's a good thing. We get human data. If we look at RNA sequencing, as we've done with cancer stem cells versus normal, that gives us epigenetic changes as well as mutations, so that expands the repertoire of types of gene changes that we get. But of course, this is focusing on mouse, and you could legitimately argue that there are differences between mouse and human cancers. With squamous cell carcinomas, they're actually quite similar, but even there's still, of course, the difference in species. So the challenge really for both of us is how do we actually sift through these hundreds of changes to really identify which ones are important, and really to stop cherry picking, which we can do, of course, but basically we're likely to overlook something simply because we don't know enough biology. So ideally what we'd like to do is to harness the power of fly and worm genetics for the mouse to be able to get our cake and eat it too, where we don't have to go to a worm. We're trying to figure out if we knock out a gene, how does this relate to human squamous cell carcinoma? And so several years ago, Boba Baranya, a postdoctoral fellow in my laboratory who's now at the Fred Hutchinson Cancer Center, and Gula Lifshitz, who was a graduate student, she's now a postdoc in Scott Lowe's laboratory, teamed together. And as we were working in the laboratory to start to knock down genes rapidly, we first tried oligonucleotides, siRNAs. That worked fine for transformed lines, and then it came to working with human culture or mouse cultured stem cells, primary cells. We couldn't get oligonucleotides in. We tried morpholinos. That didn't work either. Then we started to work with viruses, and viruses worked pretty good. And in fact, when lentiviruses came on the scene, they worked really well to put genes into the cells. And so then we wanted to get them in vivo. So Boba started to scrape the skin and apply lentivirus to the mouse skin. That didn't work very well. He tried tattoo needles. The lentivirus went into the very first cells that it saw right along the tattoo needle. Gula started working with organ transplant cultures. We put the virus in, it went and sucked up the top layer, and none of the other layers got transduced. We floated the tissue, it went into the bottom layer. None of the other layers got transduced until it finally dawned on us one day that right after gastrulation, the embryo exists the epithelium exists as a single layer that covers the embryo surface. And perhaps if we could simply expose the embryo to high titer lentivirus at this stage, injecting the amniotic sac and leaving the animal intact, and if we could do that in vivo, we might be able to simply put mom under anesthesia and inject the sacs with high titer lentivirus and get the transduction we were seeking. That remarkably worked well. This shows you 6 days later, the embryo surface, in this case marking a histone H2B RFP. We get very good transduction, up to greater than 90% in the head region. We then, once the lentivirus integrates, it's stably propagated. So now the epidermis has formed, the hair follicles have formed. Look at the dermis. It's completely untouched because it is below the surface epithelium. So this now, without having to use any fancy tissue-specific promoters, gives us tissue-specific gene expression as a consequence of lentiviral-specific infectivity, and really exploiting this property of the lentivirus. So where can we take this? Well, we could put any hairpin we want now into the lentivirus and knock down any gene that we want. And that started to give us good results. We published several different papers in the early 2010 decade, 2011, 2010, that illustrated the principle and demonstrated that once the lentivirus goes on, once it integrates and stably propagates, that all of these various different genes that are harbored within the lentivirus don't turn off, so we can monitor them into the adult. The reason for that is that the mechanisms for silencing viral genome basically is happening much earlier than 9.5 days of development. And so that worked nicely. We could knock down genes. We could knock down genetic pathways. So we started to look at asymmetric cell divisions, knocking out multiple different genes in the pathway. We could see, for instance, if we knock down a gene and it affects the p53 pathway, we can knock down p53 and see if that had anything to do with it. So we could start to do very complicated genetics that even a year ago we couldn't even dream to think about before we had this technology. But then I was dealing with a postdoc who trained in fly genetics, and a fly geneticist thinks big. And so the fly geneticist thought maybe we could take this one step further. So the first thing that we wanted to do was to see just how uniform is growth on the embryo surface. I thought for sure growth would be very variable on different regions of the embryo. So you put in a low titer GFP tagged lentivirus, I thought if you follow clonal expansion, that basically you're going to get clones of all sorts of different sizes. Turns out that that doesn't happen. Within a factor of 1 to 2 cell divisions, when the embryo is born, all the clone sizes are similar to one another. Clonal expansion is remarkably uniform, and yet if you take 100 stem cells and you put them into culture, we don't get 100 colonies of all the same sizes. Some colonies never grow, some abort, some grow big, some grow small. I think the reason is that you're subjecting the cells taken out of context to all sorts of artificial conditions. You're submerging them in growth and serum-containing medium, growth factors. They're subjected to enormous amounts of stress. You have a variety of different stress responses turned on. Some stem cells make it and others don't. But here, we're subjecting the skin surface to all the natural, systemic, heterologous, niche environment responses that these stem cells normally experience, and they grow at remarkably uniform rate under conditions where we're not subjecting the surface to a lot of stress. And so my postdoc thought, why not simply just take the entire mouse genome and set up an RNAi screen using lentiviral technology, taking the entire 78,000 lentiviruses from the library we're using. And of course, if you screen for genes that negatively affect growth, if you were to screen for that in just one animal, in a normal wild-type animal, what you get are lots of ribosomal RNA genes and lots of spliceosomal genes, lots of housekeeping so-called genes. But if you screen for differences for lentiviruses or hairpins that preferentially inhibit oncogenic growth versus normal growth, which is what you'd ideally like if you were designing a cancer drug, then effectively that worked beautifully. All the hairpin, all of the housekeeping genes basically fall out of that equation, and what you're left with then is a handful of some very interesting genes that turn out to be relevant. You have to obviously do controls, and my postdoc did 2 years of controls, setting the viral titers, setting the representation within the pooled library. You have to make sure that your coverage is good. That is to say that enough of the cells on the embryo surface get exactly the same hairpin. You have to make sure that on average only a cell gets a single hairpin. But when you do that, you can take a look at 3 different replicates, and in 3 replicates, the top 10 hits were exactly the same in all 3 replicates. If you take the top 400 hits, turns out that more than 70% of those are in the same, are coming up in the same list in all 3 replicates. To me, that was surprising. I never thought it would work as well as it did. Now, obviously, there are going to be off-target effects, but we're taking advantage of the fact that we have 5 hairpins per gene, and so what we're looking for are cases where there are more than 1 hairpin giving us the exact same differences, and none of the other 3 hairpins give us the opposite difference. And we're also then looking for dose responsiveness, and of course you also ultimately have to check to make sure that the genes that you're choosing are genes that are relevant. Our top hit, by the way, was beta-catenin, which is a well-known oncogene, and we went on to study that and demonstrate that it was physiologically relevant, as was another. And we're systematically now pursuing this. Obviously now with technology such as CRISPR, that even makes our knockouts, demonstrating our knockouts or conditional knockouts, much more accelerated for the future, which has been the rate-limiting step of basically getting this far. Once you get your hits, you have to validate them. So now I want to move in the last few minutes then to this new work that we've been doing. This work is, I'm going to cover it relatively quickly, and it's coming out in the next week or so. So I encourage you to take a look at the paper or watch for the paper if you're interested. This gets back to this issue of the Human Genome Sequencing Project and our problem with the differences in cancer stem cells. And so what we did was we said, why not simply make a smaller pool, not 78,000 lentiviruses screening the whole mouse genome, but let's simply screen 1,000 genes. Only now we wanted to look for tumor suppressors And you don't really want to look in the embryo where the embryo is growing very fast. It's a great model system to look at negative regulators of growth, not a good one to look at for positive regulators. So in this case, what we did was we used a lower titer. We used a smaller number of genes. Basically, we did a control library versus the normal versus our test library. We wanted to put it on a tumor-prone background, so we used our TGF-beta receptor conditional null mice, which don't show any signs of skin squamous cell carcinomas unless you give them an oncogenic hit like RAS. And then we simply said, well, let's just wait for tumors to develop on these animals where they don't develop in the control. And basically, let's just clone out and find out which hairpins are being selected for. So I'll give you the punchline here. And the punchline is that myosin, this is for Claire Waterman, myosin 2A turns out to have hairpins that are present in greater than 30 squamous cell carcinomas that came out of our library screen. And in fact, 4 of our 5 hairpins of myosin came out in these different tumors. And when we went back and checked them, they showed dose responsiveness. So the most tumors came with the most potent myosin. So then we tested these individually. They showed dose-dependent tumor susceptibility relative to the control. That's just an example of some of the tumors when tested individually. So I didn't believe it at this stage because we've been working on the cytoskeleton for a long time. And I didn't think myosin would be included in this kind of list. And so what we did is said, well, let's make the conditional knockout mouse, which we did. And we tested this. And sure enough, it turns out that myosin myosin 2A is acting as a tumor suppressor. And in fact, even at the heterozygous level, it actually has some potency, which turns out to be, I think, quite interesting. So then we looked in other tumors that come out. So you start with a single-layered epithelium. That gives rise to the mammary gland, for instance. And what we found are mammary gland tumors coming out. And if we stained for myosin 2A, we found a dramatic reduction. Normally, I don't have the controls to show you, but normally they'd be much stronger. You can see that these are invading tumor cells and that they basically are expressing keratin 14. So we looked at the various different tumors, used the different tumor markers. We're getting squamous cell carcinomas. So what's going on here? So I said, well, maybe it's just a tumor background. So let's do it on DMBA, TPA, classical induced carcinogenesis models. So we took our myosin knockdown animals and we started treating them with DMBA, TPA, now leaving TGF-beta out of the question. And again, we're seeing that relative to controls, we're getting higher levels of tumors, and the tumor frequency is increased as well. So then we did it on an oncogenic RAS background, same thing. And then we did it on a p53 null background. We didn't get any change in tumorigenesis. So then we wondered what's going on there. So we thought, well, maybe it's in the same pathway somehow. Maybe it's operating in the same pathway. So it was one possibility. We could test it relatively quickly. What we know is a lot about p53, and we know about how it's normally degraded in normal homeostasis, and we know how to induce p53 in the DNA damage tumor response. And so we could easily do these DNA damage tumor responses, and what we found is that actually, if you do that, what you find is that when you knock down myosin, in every case where we knocked down myosin and then did a DNA damage inducibility, what we found is that the normal increase of p53 doesn't happen. Normal increase of p53 doesn't happen. It doesn't matter when you do this, but basically we got very impressive results. If you look at p53's targets, that too doesn't happen, and we looked at a whole slew of them. And so Then I thought, well, let's just do it on the knockout. Maybe this is not going to work on the knockout. So then we took our knockout mouse cells, same thing. If you do a DNA damage response, basically p53 doesn't stabilize in response to DNA to the DNA damage response pathway, and p21 is not upregulated. So here's the whole group of them. Basically, you can show that p53 target genes also are not getting activated in the absence of myosin 2A. In this case, we're dealing with the tumorigenic models that we've generated, cells from the tumorigenic models. And so, so now we looked at what downstream is, where is myosin acting downstream. And to make a long story short, it turns out two things. One is that its ATPase domain is important. And another thing that we learned is actually if we fractionate cytoplasm and nucleus, we can show, and these are really the two main figures here, that if you treat with blebbistatin to block ATPase activity of myosin, that you can see that nuclear accumulation of p53 in response to DNA damage does not happen properly in the absence of myosin or myosin ATPase blockers. So we've looked at the pathway. Then we thought, well, maybe this is just a culture artifact. Let's look in vivo. So here's in vivo. If you take a mouse and you irradiate it, what you can see is nuclear p53. If you take the myosin conditional knockout mouse and irradiate it, doesn't happen. So I was surprised. In any case, then we said, well, what's the relevance? Is there any relevance to human squamous cell carcinoma? Because I knew my cancer friends were basically going to challenge me on this, saying, well, that's nice for your mouse, but what difference does it make if it's not relevant to human cancers? So we started to look at human cancers, and it's really interesting that actually there's a wide variation with regards to myosin expression. We did the keratin 14 controls, which apparently I don't have on this picture. But there's a wide variation in, keratin 14 is still there. There's a wide variation in whether myosin stains in a squamous cell carcinoma or not. And then we looked at 54% of all of our squamous cell carcinomas had no or very weak myosin 2A staining. 79% of head and neck cancers had no or very weak myosin staining. And then we said, well, what does that have to do with prognosis or poor survival? Turns out that the ones, if you look at which ones have no staining of myosin, and then you look at prognosis, that basically very poor prognosis correlates with low levels of myosin staining. So then we said, well, let's go back to looking at the mutations, because the people that deal at the Broad Institute are going to say, oh, if it really was important, we would have found it. And so, I mean, that's valid. I don't mind, because we have, we're relative newcomers to this. And so, so it's true that when you look, I mean, myosin is a big gene. So if you look at the p-value looks great. Then you look at the false discovery rate. And if you just look at all the myosin mutations, the false discovery rate is actually not so great. So that would make someone at the Broad Institute probably immediately going to say, well, see, I told you so. It's not statistically significant, or it's not relevant if it has a high false discovery rate. But then we said, well, wait a second. Let's take a look at what we know about myosin, and let's take a look at where those mutations reside. And then if you look at where those mutations reside, turns out that they cluster in the ATPase domains. And then my favorite is that one of these human patients has a mutation in a residue. The Jim Spudich studying dictyostelium, myosin 2A, happened to make that mutation. It's an evolutionarily conserved residue in the ATPase domain. It compromises ATPase activity in dictyostelium. So I kind of like this story, and if you like it, you can read all the details in the next couple weeks. But that's what I wanted to tell you. You're the first audience of guinea pigs, so you can barrage me with a lot of criticism on this, but I think it's really cool. So this is kind of where we've come over the last years, and we're thinking nowadays quite differently about how we study skin stem cells, and how we deal with the myriads of differences and changes that Francis Collins created for us some decade ago or so in completing the Human Genome Project. So Francis, thanks. You made us come up with new ideas about how we're going to get around these problems. I think as long as we don't get too distracted with losing the big picture for the sake of trying to focus narrowly at the bench, maybe we can come up with new ideas in the future. So these are the people. I think I've already given credit to them, to the ones responsible for the work that I talked about today. But I'd like to thank Francis again for the invitation, and thank Julie, and basically happy to take questions. Thanks.
That was terrific. We do have time for questions. There are microphones in the aisles. Please use those so that people listening over the web can hear the question. And it looks like we have one already lined up. Yes, please.
Well, what happened to baldness? Exciting work. Anyway, so what happens? Do they have enough integrins in the hair follicle stem cells so that they could participate in the normal function and growing and regeneration? You have shown nicely in the mouse that they do generate the hair follicles. But what happens in a human? Is there any major difference that does not help us to go further in taking care of these issues in human?
In humans, the stem cell, are there equivalent stem cells in human hair follicles? Or what happens to the puny hair follicles on our body surface? Could you focus the question? I'll be happy to answer it if I don't.
With what we know about the studies from yours and others,
yes,
that we could transplant the cells and grow hair follicles.
Yes.
And why we cannot translate it in human patients?
Yeah. So why can't we do that in vivo? So now you're asking a very good question. So if you can take purified stem cells and you can get hair in a nude mouse, why can't we do it in a burn patient, for instance? So there, I think what it's telling us is that we're missing a critical niche signal. And because otherwise it's very hard to explain. Otherwise you're dealing with a fundamental difference between, say, human stem cells and mouse stem cells versus a missing niche signal that is also present, that it was also lost in the burn, which in a nude mouse, the nude mouse has everything, is ready to make hair. You just have to give it a functional stem cell that can do it. And whereas in a burn patient, you're missing a lot of other parts of the dermis. So what we're now doing to try to grapple with that issue is to come up with a better understanding of all the various different niche signals and trying to identify those. We also have some pretty interesting, I think, results, which I didn't talk about, but that kind of give us some clues about what might be happening in aspects of hair loss too, which I think could also be interesting. So I think in the end, it will apply. It's just that with regards to the burn patients, I think we're missing. The experiments are telling us that we're missing one of the heterologous components that isn't there. Probably the dermal papillae, but there could be other niche signals that are critical there.
Great. Over here on this side.
Yeah, very nice talk, Elaine. By way of confirmation, Marianne Conti in my laboratory actually has knocked out myosin 2A and finds, in the tongue specifically, and finds that the tongue develops squamous cell carcinoma.
Fantastic.
But I wanted to ask you, there is a disease, very not uncommon in humans, called myosin 2A related diseases. These people get big platelets, they get defects in their hearings, and they die from glomerulosclerosis, or what's called Epstein's disease. So these mutations are all up and down the myosin molecule, including in the ATPase activity. have you been able to look to see if these patients develop any increase in squamous cell carcinoma?
Yeah, it's a very good suggestion. If it's myosin 2A, one of the things, the other thing that I didn't mention in my talk is that we've actually looked at all 3 different myosin 2s, and it turns out that the p53 effect is actually specific to myosin 2A. So it's, we don't know yet what the difference is, and I think part of the reason is not knowing exactly what the difference in mechanism is we don't know what domain is playing a role in this process. We don't know how. It could be part of the natural mechanism of actomyosin that is influencing nuclear export, which could be interesting. But until we know that, it's going to be hard to really address those kinds of questions. But I think just finding out for some of these other diseases, which are myosin disorders, whether there's actually a predisposition to any kind of tumor genesis is going to be interesting to take a look at.
Fascinating. Over here.
Hi, Len. I'm from Claire's lab. I have a question from your last part of the talk. When you see that myosin 2A mutation in the ATPase domain that is more correlative to the squamous cancer cells, do you think any transcription factor or transcriptional coactivator does get upregulated in that mutation background?
Yeah, so it could be, I mean, one of the things that we're surprised by is there's actually quite a considerable level of myosin 2A that shows up in the nucleus, and our nuclear fractions are pretty clean. And so at first I thought, oh, it's just contamination, but we kind of rigorously have been trying to test for that, and nothing is coming up with a contamination. I think it's really nuclear myosin. We have been very cautious about saying whether that's what's happening. What we know is that it's nuclear export. So we know that with regards to p53, p53 is there if we use a nuclear export inhibitor on a myosin 2A background. Then we find that now p53 can respond to DNA damage. It can stabilize, it can accumulate, and it can activate its downstream targets. So actually we're checking into whether that might have some therapeutic values, because I think that's a curious possibility. But in terms of its other potential effects, for us, the only thing that clued us in to looking at this was that as we're systematically trying it out on various different backgrounds, we tried it. I thought p53 was, that trying it out on a p53 background would be the answer, and didn't have any additional effect, which is what led us to think that maybe we're dealing with some aspect that's in the same pathway. And so it could be that myosin, if it is nuclear, is having multiple different effects, or it could be that something like an NF-kappa B type activating mechanism, that it's happening in the cytoplasm that could have additional effects. But this in and of itself, I think, is enough to explain the tumorigenic phenotypes that we're seeing. Whether it's all that explains it, I think, is a completely open question right now, and one that we obviously have a lot of things to look at.
Great. One more question.
I think the disc large tumor suppressor in Drosophila, isn't that connected to myosin 2?
Yeah, so there are a lot of suspicious characters out there that are connected. So I think, again, being interested in the cytoskeleton my whole career, I think that the whole field, I think, has kind of gotten tied up so much with all of those nuclear factors for so many years, and I think there's going to be a lot more to meet the eye than what we've seen. And I think the other aspect that I find very fascinating about the genome sequencing efforts and epigenomic sequencing efforts is that these afford the opportunity of looking for low penetrance players in the game. It may not be a common player, but it allows you to look for low penetrance players where we start from a functional assay and then look forward, as opposed to the traditional way is to look at something that's very prevalent in cancers And there you can really start with the genetics and pick that apart.
Yeah, I mean, pallidin is an actin-binding protein,
Yeah.
and that's associated with pancreatic cancer. I mean, there's
Yeah.
all kinds of stuff
Yeah.
that's been ignored at the cytoskeletal level.
Yeah, good point.
Well, fascinating presentation, and thank you for the hot new science you have allowed us to see for the first time. Please let's thank Dr. Fuchs again. And there will now be a reception in the library for those of you who would like to continue the conversation with coffee and cookies, I hope. And Dr. Fuchs will wander over there in just a moment.